Having me. It's an honour to be here. I'm the Chief Information Officer at the Wellcome Sanger Institute. I'm not a bioscientist, as will become apparent in the presentations, but I do have responsibilities for the IT that supports much of what you've heard already this morning, especially the data that's required to drive machine learning and other models. So in this presentation, I wanted to give you a bit of a sense of the importance of data to the work that we do in the Sanger, talk a little bit about the challenges that we face, the opportunities as well, talk about what is our strategy about how we manage our data. I did want to include a use case that touches on, as we've seen earlier, the fantastic opportunities in spatial and transcriptomics data. And then I've got a slide towards the end that talks, well, kind of steps back a little bit and thinks, where are we going with the technology and the big data-driven opportunities that we have here in science? Now, I did include a slide on a new project, which is quantum computing, right? We'll see if we have time to squeeze that in at the end. I know it's not necessarily big data infrastructure, but Jess and CX thought it might be worth including. So we'll see how we go. I first wanted to give you a little bit of a background to the Sanger Institute. So we're based in Cambridge in the UK. We started as an institute largely on the back of the Human Genome Project all those years ago. And since then, we've formed a fairly massive research program centered around a number of key programs within our remit. So that includes human genetics, where we do a lot of work on developmental disorders, cancer and somatic mutation. In fact, more somatic mutation than cancer, as it turns out, for early signs of disease. Parasites and microbes is a big piece of work for us, especially malaria, but we were extremely active over COVID for variant sequencing. In fact, we did the vast bulk of all the sequencing done in the UK through the period, and I think the UK, more than any other country, did so much sequencing for variant tracking. We do a lot of work in cellular genetics, and I've got a bit of a use case to talk to about that later. Tree of Life is a recent program for us where we're sequencing all 70,000 species in the UK and Ireland, quite a big undertaking. And the most recent program that started is what we're calling generative genomics, and I think that's very relevant to the conversations that we've had here today. So I've put up some facts and figures to give you a sense of the scale that we work to. The Sanger itself has a very sizable scientific operations function. There's over 250 people work in generating data, mainly from biological samples. And that kind of transition of wet lab to dry lab and what use we make of the data is quite key. I've got some facts and figures there about the IT. We currently have about 90 petabytes of data, and that's a mix of phenotypic and genomics data, and that's rapidly growing. And I'll touch on some of the challenges that gives us later. Our main workloads are large. I would call them HTC, which is high throughput computing, rather than HPC as such. So it means that being able to shift data around network-wise is quite a thing for us. So we invest quite a lot of time and money in that. I use this slide. I know it's a bit busy, but it gives a sense of some of the challenges that we face. Data is absolutely our lifeblood, if you kind of pardon the pun a little bit. And in order to maintain world-leading science, then the data has got to be present, available, and fair as it happens. So the sorts of challenges we have to grapple with are dealing with massive data, but also diverse data, and particularly thinking about new modalities. I think Kwan touched on some of that, but also about being able to share data, the interoperability of it. So as a national institute, we have quite a role there, and also thinking about multi-omics data and the integration challenges that come from that. That's quite a thing for us too. The approach that we're taking largely is that we will proactively manage data. That hasn't always necessarily happened in the past in the Sanger. But for us to get the best out of data, we need to make it easy for our scientists and informaticians to do the right thing. So A good example of that is increased use of electronic lab notebooks that help track and build up metadata, and it just happens, right? So that you're not having to expect extra effort from people. Also wanted to highlight the notion of being able to offer choice to the scientific community. So in my world, that's about offering different price points for storage, the kind of tiering that we use, and more archiving as it happens. And that's all geared up so to help our scientists get the best from the data. So given what I've said already, you probably think we love data, and I suppose we do love data. However, I would call that tough love. So if you have a look at the right of this slide, you'll see a graph that shows year-on-year growth. So this is growth you're looking at, right? I'm sure we could work out an exponential formula for it. Just in terms of the data that we're generating in the Sanger, that's it's not sustainable, right? But it is driven by us doing new science, more science, new instruments, new modalities, and the like. And one of the reasons that we have such a growth rate is, well, let's look on the left-hand side of the slide. I'm sure you've seen these before. They call it the research data lifecycle, and I picked one at random from the web. There's lots of them, but nowhere does it talk about the discipline around managing data, the deleting of it, the archiving of it, and the proper management of it. So part of my role in the Sanger is, support the scientists, new innovations, new challenges, but equally to make sure that we have the discipline in place around how we manage the data. We've just recently completed an exercise of effectively lassoing all the BAM files that we have around, and it would surprise you as to just how little compression is carried out. You can save about 30% of storage just by moving to a format like CRAM rather than BAM. So our data strategy, I wanted to touch on that, but also wanted to give you a little bit of our strapline, which is when you step back a little bit and think about what we do, largely all science is data science, whether we are generating it, whether we are collecting it, whether we're analyzing it or sharing it, that's what it comes down to. And that message is well accepted in the Sanger. We see data generation and the management of it as a key thing, a key success factor for us. So, I mean, I'm sure everyone here is roughly familiar with the kind of topics that I've mentioned there. We've adopted a kind of capability maturity model for each of those kind of topics that you might have come across previously. And the sorts of things that I would stress, particularly around skills, culture, and training on that. We do make use of data curation, and we have many data curators, but generally we're trying to get the message across that data is part of everybody's job. You know, it's quite an important message to get across. But interestingly enough, the culture is, some of the messaging has to be nuanced in as much as, yes, it's nice that PIs have personal responsibility for the data that they're generating and using, but equally, it's the project's data, it's the institute's data, it's not some kind of personal fiefdom that PIs can make use of. I would also highlight governance here. So we've embarked on an initiative to broaden out the research data sources that we use. I'm thinking particularly of what we call data diversity. And it's been great to see the welcome speeches from the Indigenous population here in Australia. But we are well aware that so much of what we do in the Sanger is mainly, like for humans, it's people of European ancestry. And we are making a commitment to getting more research data diversity, and that's quite a big thing for us. And I would also stress metadata. I'm sure you don't need to know that if you don't have proper governance and proper provenance on your data, then you're not really preparing yourself for the future. So I've talked a little bit about strategy and policy. I know it can be a bit of a dry subject, but CX and Jess suggested it'd also be good to have a bit of a use case. So this is taken from Omar Bayraktar's lab here in the Sanger, and it's largely about the infrastructure to do the kind of things that previous speakers have talked about in the way of integrating single cell and other data. So you're probably well aware of some of the techniques and technologies here. On the top left there, you can see this idea of building up offline data sources that will allow you to properly predict cell types from transcriptomics data, and we use both the Visium instrument and the new Xenium instrument. Yes, right. So if there's anyone here from WeHi, right, I guess I should congratulate you on managing to secure your Xenium instrument before we in the Sanger managed to get ours, right? I'm not bitter, but I just thought I'd make that point. And then the kind of flow through, we use Nextflow quite a lot, and you can see some of the data formats that we use there. This pipeline here that we're talking about is dealing with data entities of about 200 gigabytes as it flows through. And I just wanted to highlight the use of the ZAR format for these kind of technologies. If you're into spatial transcriptomics, ZAR is a really important format. And what it does, among other things, is it properly manages data blocks for when you want to start visualizing data. So this particular pipeline, we've used it at scale, and we run it at scale. And it's largely for doing cell type visualization and also gene expression visualization. And I think I've got a bit of a video to show. Yeah, so this is human embryo arm, and the kind of cell type identification that we can do just by using those kind of tools gives you an essence of the detail that you can get to. And again, this kind of concept of being able to zoom in and out, I suppose, delivers on the ideas behind the Human Cell Atlas, where it's about trying to get that kind of Google Maps view on tissue reconstruction. And a lot of the ability to do that, the transitions there is down to the capabilities of formats like ZAR. And you can see we get down to as far as cell nucleus. The other video is more about marker genes for us. So it's the same histological sample, but by using the kind of tools that we have here, and we've used cell-to location, which you may be familiar with, to identify the actual active genes as well as the different cell types. So we've got this running at scale. It's worth about a petabyte of data so far. And I reckon we've processed about 20 million cells, probably more than 20 million cells, and that's across 50 histology images. I'm a bit worried about pressing the button on the next one because I'm going to give you some QR codes that will take you right into the system, right? Now, it's the middle of the night in the UK, so it ought to be really quiet, so we're not going to disturb the systems too much, but I don't think I'll be very popular with my team, right, if it falls over. So do grab the codes. If you go in there, you'll be able to play around both on the left-hand side one, which is covering off just two sources of data, and then the right is for three sources of data. While you're doing that, I did want to make a few points. The kind of learning that's come out from this. This is actually a good example of being able to transfer technology from scientists and scientific community into my function, which is a central IT function. The value that we added to this kind of technology was largely about making it robust, scaling it up, adding in data and cyber security, as well as access control. It's that kind of thing. But the other point I wanted to make is that we are already reaching the limits that tools like ZAR can give us. We reckon ZAR generally copes with up to about 1 million cells or so. And we're kind of struggling after that. But you might be aware that there's a new initiative coming from the likes of Fabian Theis and Oliver Stiegel about spatial data frameworks that we're hoping will help you. Just to add to that, one of the main pieces of feedback from our clinical and pathologist colleagues was, could you make it easier to use? They don't like command line interfaces. They want drag and drop for it, right? It's okay. It's so the same the whole world over, isn't it? Right, so I mentioned this idea of, well, what does big data mean for us? And a key thing for us in the Sanger is this brand new program. It's called Generative and Synthetic Genomics. And it's basically taking two fantastic ideas and bringing them together. One of them is about multiplexed assays of variant effects. So you might be familiar with the likes of saturation gene editing or deep mutational scanning. Those kind of assays that allow you to make small individual changes right across different different genomes, different species, different assays. Linking up that kind of technology with generative AI, which consumes huge amounts of data, but if we get it right, it will allow us to establish that causal link between individual changes to individual DNA bases, and what does that mean for cells? And the third strand to this is then working that into what we call synthetic genomics, where we're looking at specifically being able to construct chromosomes, human chromosome in particular, particularly around using those models to be able to write and edit DNA very, very quickly. Conscious generative AI has got its own challenges, but you know what, we'll learn on the journey. It's a big initiative for us. So this is pretty much my last slide, and I wanted just to reflect on the kind of ideas that these sorts of programs are built on, and to what that means for us as a scientific community. So the world I come from, people talk about digital transformation a lot, and that applies equally to us as scientists, where we ought to be exploiting, you know, the benefits of big data and the models, not just for doing science, but the way that we do science as well. And the classic example I've quoted here, I'm sure you all do it, about using large language models to help with literature searches and things like that, help speed up the process, reduces the time to science. But there's a bigger story there. And over the next number of years, you'll start to see more and more foundational generative models starting to play in this space. I've included an example which is you know, more directly relevant to the Sanger, which is about getting an end-to-end digital infrastructure that takes us all the way from biological samples through to the you know, secondary, tertiary analysis that we do, linking up the likes of LIMS systems, electronic lab notebooks, Jupyter notebooks, so that you get a totally end-to-end perspective for shifting data. I think we'll see increasingly sophisticated models coming through. So if you've got reference data, or you're working on a cell atlas, or even like the UK Biobank, those will be more and more AI ready, and you can expect to be asked about, is your data AI ready, and what that entails. And as you saw from the use case I had there about data science for visualization and pattern finding. So this is the new world that we're in. And if you're familiar with the work of Jim Gray, they call this the fourth paradigm, right? This is the fourth paradigm of scientific discovery, and it's happening now. So by exploiting the data that's available, by exploiting the tools that come along, then the whole nature of the way that we do science will change. And it's a very exciting time, actually. It's a very exciting time for a community like this, where previously scientists would automatically go straight to wet lab. They'll more and more be using foundational models, so that's important for us. So I think that was my last slide. I just wanted to thank you for your patience and listening. Thanks to my team, who are fantastic, and also thanks to all the scientists who have helped shape the journey. So thank you. Do I have time? Right. Okay. Okay. I'm going to do an encore now, which is. Okay, right. So one of the projects, it's not really so relevant for big data and infrastructure, but I did want to talk about it if I had a chance, which is a new project that we're starting. This is in collaboration with the University of Cambridge and the European Bioinformatics Institute, and it's about quantum computing. Now, if you'd have asked me about a year ago about quantum computing, I would have just dismissed it out of hand. But the field is moving on quickly, and it's worth everyone just taking a little bit of time to, you know, to appreciate what the systems can do. Now, well, certainly not in my lifetime. We're not going to see quantum computers on your laptop, right? That's not the way it works. Generally, yeah, I was disappointed too. But generally, the way it's going to work is that when these devices appear, and trust me, they will appear, they are going to be like a GPU farm, right? That's the way it's likely to work, where you'll have classical computers preparing data workloads that it will push out to the quantum computer, and the quantum computer comes back quickly with the results. It's a totally different paradigm for computing, and I've had the pleasure of seeing some very senior bioinformaticians struggling with the ideas of what it is in the quantum world, and that's not just about the terminology and the vocabulary, but some of the concepts as well. So, for example, in the top right there, you'll see this notion of a qubit, a quantum bit, and a quantum bit, sort of like a bit in digital classical computing, except that it has the power of representation. So, for example, in theory, at least, you can hold an entire human genome on just 32 qubits. Right? Of course, the problem with quantum is that when you look at it, when you actually measure it or you want to use it, it collapses, and it collapses down into just this number of qubits. I put a little graph on the left there. This is from NIH, and it shows the demand that we have for compute in our field for genomics as compared with Moore's law, right? And the metric they're using is the cost of the human genome, which we know is now less than $1,000 per genome. And Moore's law just can't keep up with that. And all the presentations that you've had throughout this conference about the massive amounts of data being generated, we need to be able to compute. Now, quantum computers are not the answer to every problem, but what they are good at is, I would call them low data, high compute problems. So you hear about them being used for the likes of factorizing prime numbers, that kind of thing. So for the work that we do in the Sanger, we see that as quite a good link into pangenomics, if you're familiar with that. So in pangenomics, you're using a variational graph to store the diversity of a population, and it requires massive amounts of multiple sequence alignment. And if you're not careful, that's a n to the power m problem, right? With heuristics, you can get it down to n times n, which I suppose is progress. But I've put here a graphic taken from the recent paper, I think it was in May, of the first human pangenome. I think they had about 50 separate individuals. So that's a good problem to go after because it is relatively low data, but high compute demand. And what we're going to do is we'll do it step by step. I have a fondness for Phi-X174 bacteriophage, if you like horrible bacteriophages. We're going to sequence that. We're going to process it on a quantum computer and just get a sense of what it is to do, genomic assembly and the like on it, and then progress through SARS-CoV, and hopefully eventually we'll get to the human genome. This is very speculative, I appreciate that, but it's something that we are quite keen to progress on, and I suspect we'll learn as much about genomics as we will about quantum computing as we get into them. Wish us luck. And if anyone wants to talk about quantum computing, we're really keen to get lots of partnerships and collaborations around this. It'll be great fun. It really will. Okay, I think that's it from me. I'll really shut up now.
Incredible. Thank you so much, James. That was so fascinating. I believe we have already some questions lined up. Let's go.
Given I was sitting very close to the mic, I felt like asking a question. So about the data volume you showed, like 90 petabytes, I think. Is it like a part of a publicly available archive, or is it internal to the organization?
Oh, right, yeah. So that's mainly Sanger-generated data. We have a very close relationship with EBI, and we pump huge amounts of data into the EBI, ENA and EGA, if you know about them. But generally, that's all Sanger data. We do host. I think.
I was quite interested in it was almost like a throwaway comment. Each sort of data set by the sounds of it. I wonder if you could talk to that. Is that just wet lab, or is that
Yeah, sorry about the throwaway comment. We have notebooks to cover as broad a range as we can. So I mentioned sample management, the DNA extraction, and the labs, as well as the big sequencing facility that we run, all the way through to the actual capturing of results and being able to properly publish them within the organization. So that sits on top of our LIMS capability. But in addition to that, I mentioned this idea of taking that into the secondary and tertiary analysis, so tools like Jupyter Hub, so that we can get it working end to end. And the nice thing about ELNs is that they automatically capture a lot of metadata, so it makes it easier.
Thank you, James, for flying down here to give us such a great
So we're all looking for the right options going forward. For the cloud, we do use it. It tends to be for niche applications or situations where it's allows us to respond very quickly. So for the UK Biobank work, and Sanger did a massive amount of sequencing on that. That largely it came out of the blue. It's something that we had to turn around very quickly and progress. So for certain applications where it's rapid turnaround time, or we are working with certain collaborative communities, then we're more likely to use the cloud. And we have different tools to monitor usage, in particular monitor cost, and a lot of that ends up on my desk for what we're doing about it. I should say the scale of what we've got, it would be very, very expensive in the cloud to move it across.
Just as an aside, IBM just announced a 1,000 qubit computer this morning.
Lovely.
Yeah. And my actual question, I think you partially answered, is what proportion of the Sanger's data do you think gets deposited in ENA or other public repositories?
Yes. So all of the human genetics work that we do goes through the Tree of Life program. Do pass data into EBI, but they're quite an interesting story. They've also set up a means of what they call genome notes that they publish independently. I think it goes through to Wellcome, so it's worth having a browse to download that. For cellular data, not so much. But then I don't think EBI is necessarily geared up for so much of that. But certainly for human genetics, pretty much everything.
And what about all this new spatial stuff? Is it, I don't know.
Yeah.
I'm in parasites and microbes, so I don't really know that.
Yeah. So EBI are setting up an image archive, Virginia Ullman, and it's fairly new, but I think they're seeing it as will be a key repository for all of us to make use of.
All right. Thanks.